It's really nice to see this many people here in person. We do encourage use of the entire space, and we haven't actually done that. So if you wanted to spread out a little bit, our hospital epidemiologist would be more than happy if you were to do that. Welcome to the 25th Annual Astute Clinician Lecture. I'm Jim Gilman, CEO of the NIH Clinical Center. Today's special lecture is an NIH Director's Wednesday afternoon lecture series event hosted by the Clinical Center. Dr. Tabak, currently performing the duties of the NIH Director, is unable to join us today due to other commitments. The Astute Clinician Lectureship is established through a gift from the late Dr. Robert Miller and his wife Haruko. It honors a U.S. scientist who has observed an unusual clinical occurrence and by investigating it has opened an important new avenue of research. Now I welcome Dr. Stephen Holland, who will speak on anti-cytokine autoantibodies, causes, concomitants, and complications of infectious diseases. Dr. Holland earned his MD from Johns Hopkins University, where he remained for his internal medicine residency, chief residency, and fellowship in infectious diseases. He came to NIAID as a National Research Council Fellow in 1989 to the Laboratory of Molecular Microbiology, working on transcriptional regulation of HIV. In 1992, he joined the newly formed Laboratory of Host Defenses, shifting his research to the host side with a focus on phagocyte defects and their associated infections. Dr. Holland is an NIH Distinguished Investigator, NIAID Scientific Director, and Chief of the Immunopathogenesis Section. The section utilizes a bench-to-bedside model for a fully integrated approach to infectious disease, incorporating the molecular genetics of the host and the pathogen, as well as mechanisms of pathogenesis, together with clinical appreciation and investigation to gain new insights into both mechanisms of action and avenues of therapy. Dr. Holland has received the American College of Physicians Award for Science, the Boyle Scientific Achievement Award of the Immune Deficiency Foundation, the American Society for Microbiology Abbott Award, the Erwin Nieder Award of the Association of Medical Laboratory Immunologists, and the NIH Distinguished Clinical Teacher Award, among other honors. He is recognized globally for his scientific expertise and is a true role model as a caring, compassionate clinician to all of his patients. Dr. Holland exemplifies the traits for honorees upon which this lectureship was created 25 years ago. And now, Dr. Holland.
Thank you, Jim. There are few opportunities more anxiety-provoking than speaking in front of your home crowd. And I will try to keep that under control as I go through. Let me just begin by saying that it's an incredible honor to be here, and it's an honor because I have the opportunity to present work that is not entirely mine by a long shot. And clinical medicine really is a team sport. And you know, there are the people that make the team go at every level, and I'm not going to go through and name each one here, but just point out that it's impossible to do these things on your own. So there is no one astute clinician. There are astute compilations of investigators, and it is a real honor and pleasure for me to be a part of that. I just want to mention briefly how I got here, because as I was reflecting on that, it was a little amusing. In Baltimore, I had the privilege to serve as a house officer under Victor McCusick, and during that time I got really interested in infectious disease. Maybe I was a little interested before, but and then John Bartlett was just an inspiration at every level of infectious disease training. And Diane Griffin had a unique ability to integrate how infectious disease and laboratory science came together. I left my residency and my fellowship, my clinical fellowship, to work in the lab of Tom Quinn and then Tom August there in Baltimore. And all the while, began to think that there might be something else that I could do. My father kept on saying, you should go to the NIH. No, you should go to the NIH. No. And finally, 3 times and I was out. So I said, okay. And I worked in the lab of Mel Martin, in the lab of Sundarajan Venkatesan, who was a PI in the lab at the time. And as I was there, on Friday afternoons, because back then we did have opportunities to enjoy personal time with colleagues in the lab, my friend from fellowship, Skip Francis, shown here as the winner of the Mighty Old Ducks season 2022 with the critical championship logo. Skip would come by and say, Steve, there's this guy you got to go talk to in the clinical center because he's got a job that you would like. I was in the lab, I was doing basic science, I was heading back to Baltimore, my wife lived in Baltimore and so on, and I didn't. And Skip came by a second time and he said, did you go? I said, no. And he really, thrashed me and said, that was crazy, you got to go talk to this guy. And so I did. And I went to talk to John Gallen, who at the time was becoming the scientific director of the Institute. And he described to me this fantastic opportunity to have a laboratory 50 yards away from the inpatient unit and to work on anything I wanted as long as it was neutrophils. And I said, well, neutrophils, those aren't the smartest cells, are they, John? He said, yeah, but they're my cells, and they're really cool cells. And after you work on those, then you go and you find your own disease. And he meant like, discover your own disease. I thought, this is crazy. But, it was a chance to do something that sounded pretty exciting. And so I did take that challenge, and I have really been pleased that I did, and I've been enormously grateful to John and to so many people along the way. So here's the story. The number of immune deficiencies that are recognized has been growing by leaps and bounds. And you can see here across the bottom, going back to the 1980s, but here in the last few years, the number has really taken off. This, of course, reflects the change in DNA sequencing that makes this also possible. But what's the real rate? That's the rate in cases. What's the real rate worldwide? And so when you go and look worldwide, it looks like the rate, at least in this country, is somewhere between 5 and 10 cases per 100,000 have primary immune deficiencies, now known as inborn errors of immunity. I point that out because in contrast to that number, what's the rate of tuberculosis in this country in the last couple of years? It's about 2.7 per 100,000. That is the prevalence. I get it, there's a difference between incidence and prevalence, but the prevalence of the primary immune deficiencies is several-fold higher than the rate of tuberculosis. And yet think about, if you're a regular doctor out in the world, how often do you look for TB, and how often do you stop and look for primary immune deficiencies? And one of the categories that's growing most rapidly now is the group of what are known as phenocopies. And these are in part due to somatic mutations, which I won't talk about, but autoantibodies to cytokines in particular. And those are shown here on the right side of the slide, and I'll go through some of this in painstaking detail over the next couple of hours. So we're only going to mention a few of these here, interferon gamma, IL-23, Gm-csf, and interferon alpha, but I hope I can give you a flavor for why these are important and why you might want to know. So my interest in this starts here with mycobacterial disease. And now, 140 years ago, Robert Koch invited people over to his house, and he showed them, under the microscope in his kitchen, the organism that we now know as Mycobacterium tuberculosis. And he tried to persuade them that this organism was, in fact, the cause of the disease that everybody knew as a clinical entity. Of course, they didn't believe him. And they said, no way, itty bitty, no way. And that's why we have these things now called Koch's postulates, trying to prove to us that things really are what they seem, they cause it, they can be recovered, and so on. The other critical consequence was everybody said, never am I going to have dinner at Bob Koch's again. And there are consequences to everything. So this is M. tuberculosis, 140 years ago. But despite the fact that we've had generations to reflect on it, we're still a little unclear why people get sick and why most people don't. And so in the 1980s, with the advent of the AIDS epidemic, it became clear that susceptibility to tuberculosis and non-tuberculous infections was a hallmark of immune deficiency. And so coming into the 1990s, many laboratories, my own included, decided to pay attention to people who got severe non-tuberculous disease, and these are all grouped together under the rubric non-tuberculous mycobacteria, or NTM. And we said, we'll go look at those infections, and if we take HIV out of the mix, maybe we can find underlying susceptibility causes. And there are three major types of mycobacterial disease. I'm not going to talk about the more common ones, skin disease, caused often by pedicure parlors. The key here is don't shave before your nails are done. I'm not joking. And then lung disease, which is far and away the most common cause of mycobacterial disease in this country, but one about which we still understand painfully little. But disseminated disease is where we really have our greatest knowledge base. And this is like disease, I think, in most categories. We know the most about the rarest, most unusual presentations. They're the easiest to dissect. And what we know in general is that mycobacteria infect macrophages. That leads to the production of a variety of cytokines, including IL-12 and IL-23. Those lead to the production of interferon gamma by T cells and NK cells that then stimulates its receptor that is present on all nucleated cells and leads to the activation of intracellular pathways that lead to the killing of mycobacteria through mechanisms that are still, frankly, not clear at all. But we know that this is the right pathway because we've got Mendelian mutations in a host of these genes, as shown here, and there are others not shown, that are clearly responsible for severe disseminated mycobacterial disease. Not usually tuberculosis, typically BCG, which is the provocative agent used throughout the world, not here, of course, used as a vaccine, but turns out to be a very effective way to figure out who's really got susceptibility. Now, this pathway is referred to as Mendelian susceptibility to mycobacterial disease. And although that is true, I think it's important to remember that it's not just about mycobacteria. And these patients with these mutations and these pathways, yes, they get infected with mycobacteria, but they also get infected with gram-negative rods, gram-positive rods, fungi, and viruses. And these infections are just as severe and debilitating as the mycobacterial ones, although were recognized a bit later. Now, coming to the critical part of the story, most of these infections are recognized in childhood. That's particularly true if you're administering BCG vaccine. It's given on day 1 of life in most countries. And so, you get inoculated early. That leads to disseminated disease early. And those diseases show up early. But if you're in places that don't use BCG, then it can be a little bit different. And so this lady was really a critical introduction to this for me. So a 60-year-old woman, born and raised in Vietnam, survived the war, came out through the refugee camps and on the boat lifts, and came to the United States in the 1970s. Perfectly healthy, successful family, successful businesswoman, and then at 59 began to lose weight and to be quite ill. And eventually was referred here, at which point she had these lesions on her chest wall that were due to osteomyelitis in her bones rupturing out through her skin. And so the question on referral was, number 1, how do we treat this? And number 2, why did this happen to her at age 60? That's young, but she's not an infant. So what is going on? So our question as clinicians was, where is the lesion? And the simple answer was not any place that we knew to look. So our problem was that this looks like a genetic disease, it acts like a genetic disease, but it's coming on late in life. Not that genetic diseases can't, but it's not what they usually do. And in her in vitro cellular behavior, once we washed it free from her plasma, was perfectly normal. So to cut a long story short, what she's got is an autoantibody against interferon gamma, the ligand, which is completely neutralizing and therefore blocks its ability to interact with its receptor, which then therefore mimics the defects in the receptors and its downstream pathways that we had recognized previously. So why is that going on? So here's a 60-year-old woman from Vietnam showing up in the United States with this problem. At the same time, papers were being published that gave a hint about this. So here in 2004, a 25-year-old Thai woman identified in Germany with Mycobacterium coloni sepsis, followed by Burkholderia gladioli sepsis, which was fatal. In the same year, a 57-year-old Filipino man identified in Great Britain who had tuberculosis, rapid-growing Mycobacterium M. coloni, and then autoimmunity and died. And at the same time, we were identifying this cohort, of which this woman was a member, 6 women, all from Southeast Asia, all in the United States, all with severe disseminated disease, although none proved fatal. So the question that we tried to address was, these were all Southeast Asians outside of Southeast Asia. All of them were afflicted by IgG autoantibodies against interferon gamma, as well as other opportunistic infections and a lot of autoimmunity. These were bad problems. So how were we going to figure out what was going on? So we reasoned that if this is happening in Southeast Asians outside of Southeast Asia, it would make more sense to go back to Southeast Asia and see what was really happening there. So I was lucky, and one of the great perks of reviewing papers for journals is that you often see things before other people do, and sometimes you see things that you might not see if you were just thumbing through a paper journal, not that you do, or if you were just skimming through titles. And I had a chance to review this paper by Plonchan Chetsoksat, about 59 patients in Thailand who had severe opportunistic infections, none of whom had HIV. And you can see here she was from Khon Kaen, where these patients were accumulated, but they came from all over Thailand. And you can see all of their opportunistic infections. So these are infections plus non-tuberculous disease. So again, salmonella, cryptococcosis, all the infections that I showed you earlier that occur in patients with the genetic defects. And so we reached out to her, and Sarah Brown in the lab developed a protocol to go to Thailand and Taiwan, where these cases were also identified, and try to figure out what was going on. And we developed an assay that I'll show you in a second that we decided to apply across 5 different patient groups. And those groups were those with non-tuberculous disease only, disseminated non-tuberculous disease, other opportunistic infections with or without non-tuberculous disease, disseminated tuberculosis, pulmonary tuberculosis, or normals. And we had an assay to look for interferon gamma autoantibodies developed by Peter Burbello, shown here incognito because I don't want you taking up his time. We still want to collaborate with him. But Peter had this assay to look at 41 different cytokines through something called LIPS, which is an absolutely brilliant assay that allows for high throughput and high sensitivity. And showing you only the interferon gamma data here, what you see is that patients who had non-tuberculous disease or other opportunistic infections had high titers of antibody against interferon gamma, whereas those who had disseminated or pulmonary tuberculosis did not, nor did blood bank donors. And this was really quite striking to us. That was like 90% of people who had opportunistic infections had antibodies to interferon gamma. What were the other opportunistic infections that these people had? Exactly the same ones I showed you before. Salmonella, Burkholderia, crypto, histo, Talaromyces, as well as severe viral infections such as varicella zoster. What do they look like? This unfortunate man had both disseminated Talaromyces marneffii, used to be Penicillium marneffii, as well as Mycobacterium fortuitum. This woman had Salmonella lymphadenitis. You can see this young man had cryptococcal osteomyelitis, and this woman severe mycobacterial skin disease. So these autoantibodies that recognize interferon gamma and neutralize it mimic the genetic disorder of Mendelian susceptibility to mycobacterial disease by neutralizing exactly the pathway that we knew was important. But why? Why should it be happening? Why should it be happening in mostly this group of people? And why should it be coming on later in life? So in some beautiful work from Chunlong Q's group in Taiwan, they reasoned that this is happening regionally in people of one certain ethnic background. And one of the things that ties together ethnicities is HLA type. And so they went through the HLA types in these patients, wondering, are there any that they have in common? And what they found was that there were these two HLA types, DRB1 and DQB1, that were very highly associated with people who had autoantibodies to interferon gamma. Why would that be? And so they looked at the binding groove that these haplotypes caused, and what they found was that it recognized 9 amino acids. And those 9 amino acids were the terminal amino acids in interferon gamma, shown here in red. Why is that important? Because those same 9 amino acids, shown here, are identically mimicked in a gene in Aspergillus. And there are many things that are similar. This one is identical. Some of these are very similar. And so the idea was that there is an HLA predisposition to recognizing a peptide that allows for the development of an autoantibody. And as shown here, the idea is that people would get exposed to Aspergillus. They would develop antibodies to the Aspergillus protein. That antibody would cross-react with interferon gamma, leading to complete neutralization. And they proved that this is a plausible explanation because when you deleted that epitope from interferon gamma, it restored its ability to signal. A good explanation that helped to verify that what we need to do then is to go after those antibodies. And so as is true in so many things in medicine, once you have a cause, you at least begin to know where to go to exert an effect. And so early on, we recognized that this was an antibody problem and decided that we would use rituximab to try to reduce it. And you see here the effect of rituximab. We have the starting titer and then the after rituximab titer. In this one person, the woman whose picture I showed you, when we stopped therapy, her antibody levels came back, as did her B cells. We re-implemented rituximab therapy, and she went back down and cleared her disease. There are, of course, patients who don't respond to therapy. I think there are patients who don't respond to therapy in every disease I've ever seen. There's always variation. And so once you decide it's an antibody-mediated problem, then the question is, how do you go after doing that? And so in work that Krista Zerbe has done to use then the downstream approaches, in this case bortezomib, a proteasome inhibitor active in plasma cells, to reduce the titer of antibody in this patient so that she could clear her disease, as you see here, going from an abscess to none. And in those patients that either could not tolerate or did not tolerate bortezomib, then to go after plasma cells directly using this drug, daratumumab, which, as you can see here, this woman who had large abscesses that resolved with therapy. Finally, how do you sort this out? If you're not in an institution that does this assay frequently, and most of us are not, you actually already have the data almost all the time. And that is shown here in the quantiferon assay. So just remember the quantiferon assay, that's the peripheral blood draw that you use to decide whether somebody's got antigen-experienced T cells that respond to mycobacterial epitopes. It is based on the generation of interferon gamma in response to those epitopes. And so you have 4 tubes when you do it, which is why it's so common that it gets rejected by the laboratory, because they get filled sometimes too much, too little. You know, you need the 3 little bears thing. But here you've got the nil tube that has no interferon gamma in it. the antigen 1, antigen 2, or a mitogen to activate the cells to make sure that they're alive. And you see here typical values. If you've got a TB antigen response, then you say, okay, that person's been exposed to TB. If you have no antigen response, but the mitogen is positive, then you say, okay, that person's not been exposed to TB. But you have the mitogen tube in there to make sure that the cells are alive and responding. And in the case when the mitogen tube does not have interferon gamma detected, that gets called indeterminate. And it can be indeterminate because the cells died, or it can be indeterminate because there is an antibody that blocks the signal. And so if you've got a quantiferon assay on your patient, you already have an idea whether they've got autoantibodies to interferon gamma. It's not 100%, but it's pretty good. And it's a good approximation of what is going on. Finally, these antibodies, of course, turn out to be more complex than we thought. And they're more complex because they recognize many different epitopes. I told you a story about a single epitope that tells a great story. But biology doesn't often stay within those narrow confines. And it turns out there are several categories of antibody that are generated in this disease. And to summarize it schematically, there are at least 3 types of antibodies. Those that recognize the interferon gamma alone and prevent it from binding, that's easy. Those that recognize the interferon gamma as it is bound to one of the receptors and prevent the receptor from heterodimerizing and signaling. And then those antibodies that generate ADCC, that block ADCC, that is part of what they think is being used in the activity of interferon gamma for some of these cells. So 3 different types of antibodies that are involved in the generation of these problems. So these anti-interferon gamma autoantibodies are a fascinating set of manifestations of autoimmunity. They are genetically determined in part because there's an HLA association. There is a strong gender predisposition, not in Southeast Asia, but once the patients leave Southeast Asia, it's overwhelmingly the women who get sick, not the men, even though they're all born in the same areas. It's about 10 to 1, female to male, outside of Southeast Asia. And then there's a strong geographic component, because people born in this country are no more susceptible to it, even of the same ethnicity, than others, as far as we can tell. There are multiple opportunistic infections. There's a clear association here with the HLA types, and it is therapy responsive. So this recognition of antibodies against interferon gamma makes a lot of good sense. So it certainly was reasonable that we started to look for other antibodies in the same pathway that might be important. But there are questions that remain about interferon gamma. How many ways can you impair function? I showed you 3 that are known. There may be others. Which titer should we be measuring? If there are several different ways to bind, maybe measuring just those that bind to the epitope on interferon gamma is not enough. Are they the right markers? What's the right environmental exposure? Why is it so localized to that region? And do we need to treat everybody? These are questions that we're still grappling with. But we talk about the interferon gamma antibodies. As we go around our circle, we thought that we had a good idea to start looking for autoantibodies against IL-12. It made a lot of sense. And when we identified a woman also from Southeast Asia who had 4 years of bacteremia with this unusual Burkholderia, we really thought we were onto something. And she had an antibody against IL-12, which is right there in that same pathway. It made a lot of sense. But of course, this is where having new people come into the lab and tell you what they think is often educational. And so Aristine Chang came into the lab from Taiwan and said, no, I don't believe it. The anti-IL-12 antibodies are too common. They're very common in thymoma. And they don't seem to do very much in most cases of thymoma. And there's never been another report of an anti-IL-12 autoantibody causing severe disease. So she said, you know, I really think you missed the boat here. It must be something else. And I'm here to tell you she was right. I was wrong. And what she realized was that these antibodies, yes, they bind IL-12, but more important, they bind to the P40 component of IL-12, which is shared with IL-23. And this antibody can be purified out of plasma. It's got high binding activity, and it blocks the ability of IL-23 to activate STAT3, which is simply a readout of this pathway. So why is that important? Because IL-23 is required to teach the mucosal-associated invariant T cells how to make interferon gamma. And those cells are responsible for patrolling everything from the mucosa of the lung to the mucosa of the gut and elsewhere. And these antibodies completely block the ability of mucosal-associated T cells to produce interferon gamma. So once you recognize one, this is the problem of uncertain reinforcements. It does make you keep searching for more. And as we began to think, well, could this be involved in other infectious diseases, Aristine started to collect a variety of samples of people with severe disease who had not had etiologies identified. And what you see here are the rates of IL-23 autoantibodies in patients with severe invasive infections. There are a lot of them in Thymoma, and there's an overlap with IL-12 because they share a component. But there's also some other disseminated infections, as well as some, these are mycobacterial infections, and these here are fungal infections. I'm just going to show you one of those, a patient that Mihalis Leonakis has taken care of and cured successfully of her severe brain infection. You can see here a CNS disease with Cladophilophyra bantiana, which is commonly known to be bad. And that infection is not easy to cure, and it's always been unclear why people get it. In this woman's case, again, a younger woman from the Philippines, what she had was a high titer antibody against IL-23, only found after the fact. But you can see, once again, she's got antibody that is neutralizing, mostly an IgM antibody in her case, as well as fully prevents STAT3 phosphorylation up to high concentrations and blocks the activation of mucosal-associated invariant T cells. So now we've got two components here, interferon gamma, IL-23, and the IL-23 antibodies, which are not yet published, I think are quite interesting because they're subtending the susceptibility to a variety of infections, very much like interferon gamma. Mycobacteria, bacteria, fungi, viruses, and they are common in Thymoma, but they're separate from the IL-12 antibodies. Okay, so moving away from the interferon gamma pathway, as we were exploring that population in Thailand, we realized that there were people who had disseminated opportunistic disease who didn't have anti-interferon antibodies. These are the group that did. These are the people who didn't. This man here had severe cryptococcal disease involving bone and the CSF. So why should he have disseminated cryptococcal disease? And he didn't have an interferon gamma antibody. What he had was an antibody against Gm-csf, a cytokine that is made by many cells and responds, and many cells respond to, including alveolar macrophages, T cells, and many other somatic cells. And this antibody against Gm-csf, many of you will recognize as the cause of pulmonary alveolar proteinosis, the condition that causes this syndrome known as crazy paving. And it's often associated with the filling of the alveolar space with fluid because it inhibits the ability of the alveolar macrophage to metabolize alveolar content. And so it cannot ingest it, cannot digest it, cannot clear out the alveolar space, and it leads to progressive obstruction and loss of breathing capacity. But these antibodies were not associated with that, at least not primarily. They were associated with, in this case, cryptococcal disease. And as we went back through all of our cryptococcal background, we went to Jack Bennett, who had many samples from cryptococcal meningitis in the freezer from many decades of study of this disease. what we realized working with Junquan Chung is that these autoantibodies to Gm-csf were responsible for around 10% of cases of cryptococcal disease. But not just cryptococcal disease, it was Cryptococcus gattii. So for those who aren't cryptococcal nerds, there are two major forms of Cryptococcus. There's neoformans, which is what you see mostly in HIV-infected patients, and gattii, which is the quote, immunocompetent form of Cryptococcus. And this is only really associated with the C. gattii. And then, of course, as you keep going down this path, it turns out that, like is true for so many things, the more you look, the more you find. And this man was driving through Northern Virginia when he had a seizure and was taken to a hospital where they noticed this brain lesion and said, this has to be operated on. Absolutely the right thing to do. For those of you who are infectious disease specialists, you'll know that the most unexpected part of this was that it was cultured and that it turned out to be nocardia. And after the appropriate treatment was done for nocardia, they began to wonder, well, why should a man who is in really robust health have gotten severe CNS nocardiosis? And so they referred him to us. We were really excited to see him. And Lindsay Rosen in the lab took on the question of what's going on here? Well, we early on realized that he too had autoantibodies to Gm-csf. Those antibodies inhibit the ability of the macrophage to respond to its own Gm-csf that's stimulated by the infection by nocardia. So it blocks the autocrine pathway for macrophage activation. And then finding one case doesn't really help. You have to find more. And so we went around the world to our colleagues who had patients with disseminated nocardia, and sure enough, there were many cases out there. Okay, 5, but for us that's a lot. It's about as high as I can count. And 5 cases more of disseminated nocardiosis in the setting of autoantibodies to Gm-csf. Now, okay, so I mentioned we've now got 3 different categories of what Gm-csf autoantibodies can do. There's pulmonary alveolar proteinosis, cryptococcal disease, and nocardia. Are those different diseases, or are they the same disease? And are they the same disease with different manifestations? And so Hélène Salvatore, who was a visiting fellow in the lab, took all of those cases and said, let's compare what they do. And the answer was, they're not very different. There's a little bit of difference here, but it's not very significant in terms of overall titer of antibody. And so then we started to look at individual cases. And this is where it gets weird. So in this patient, who Krista Zerby is taking care of, he had severe pulmonary alveolar proteinosis, never had an opportunistic infection at that point. And you can see here when we look at something called the EC50, that's the concentration of Gm-csf that's required to produce 50% STAT5 phosphorylation. That STAT5 is our readout for this. It turned out that the change in his neutralizing capacity wasn't very much over the course of years of treatment from 2016 to 2020, despite the fact that the binding titer went down considerably. That suggests that we are measuring 2 different things: what binds to the ligand, what blocks the signal. And it harks back a little bit to the multiverse of antibodies that we saw in interferon gamma disease. So that's a little bit distressing. It means that we may not be measuring always the right thing. We may be in the right neighborhood, but maybe not in the right house. And when we go back to our patient who had the brain disease and ask what happened here, it's even a little bit more confusing. Because he was in the military, we had samples of his blood dating back to 13 years before he presented with his brain abscess. And Hélène was able to show that the difference in titer over those years, from before infection to his subsequent development of severe pulmonary alveolar proteinosis, didn't change very much, even though we were able to watch his titer change a lot. again suggesting that we need to refine what's the target in order to really understand what's going on. So these anti-Gm-csf autoantibodies cause a variety of confusing things: pulmonary alveolar proteinosis, severe cryptococcosis, and severe nocardiosis. Many of these patients present with infections and then go on to develop pulmonary alveolar proteinosis. So when you identify them once, you still have to follow them longitudinally to make sure you know what's happening. Some of these patients are Gm-csf responsive, especially in aerosolized Gm-csf, perhaps because it can achieve a higher concentration to overcome antibody in the alveolar space, perhaps for other reasons as well. Okay, coming into the end zone, so you don't have to wake up yet. But the last thing is to mention about what's going on with COVID. And in beautiful work that has come out over the last several years, the team of Gigi Notarangelo, Helen Su, and Jean-Laurent Casanova has collected a lot of samples from around the world to try and understand what is going on both genetically and phenotypically in this disease. And through a series of papers, starting with this one in Science in 2020, they identified about 3% of patients who had monogenic lesions that were associated with severe disease. There's been some controversy about this, but I think that there's a lot of accuracy to the fact that if you've got lesions in genes that impair the ability to either generate interferon alpha or respond to it, you're likely to have a problem handling viral infections. And so early on, they recognized that there were problems in this pathway. But that didn't account for very much of a disease that had a huge impact on all of us. And so working with Lindsay Rosen, we began to look at are there autoantibodies here that might be important? And the answer is yes. And in about 10% of patients with severe COVID, there were autoantibodies against the type 1 interferons, both interferon alpha and interferon omega. And those autoantibodies are neutralizing and high titer. And that really, again, reminding you that there are genetic lesions, which are rare, but then these things that we're talking about, these autoantibodies, are likely to be more common. Making it even more confusing is that they're dynamic. And in work that Ilana Shaw and Helen Su and Lindsay Rosen did, they were able to show that these antibodies are high when people come in sick, but if you follow them, which not everybody wants to do, if you follow them over time, in many cases, these antibody levels fall back down toward the normal range. As you can see here, within 2 to 3 months of presentation with acute disease, people were pretty much back to normal. So now we've got a series of problems. We've got antibodies that are present when people get sick. We've got antibodies that fall after people get better. And how do we put all that together? And one of the important points about COVID is that it is really a ripple in our pond that has lots of consequences. And sometimes once you light the fuse, you don't know what is going to happen. And in some elegant work from P.J. Utz's lab in San Francisco, they've looked at what happens to people after COVID infection. And I'm not going to go into the specific targets here, but if you look at people who are post-COVID as opposed to people who did not have COVID, you see a marked increase in the number of autoantibodies present. Now, not all of these are neutralizing, not all of these are clinically important, but they're all there, and they all came up in the post-COVID setting. And so as we try to understand why is this such a dynamic situation, we need to be thinking, what is it that COVID is really stimulating? And as we move into the holiday period, we will all have a chance to be contemplating autoimmune responses. Finally, these things have real-world implications. And this is just one of the more stark examples, a completely healthy 40-year-old man who decided he wanted to go on safari. And he got the yellow fever vaccine, and a week later showed up in his local emergency room with yellow fever. And he had hepatitis, he was sick, and he, being a physician, said to the team in the ER, I got yellow fever. And they said, yeah, you're right. And when they did an x-ray as part of his standard ER evaluation, they said, oh, you know, either you've got a second heart there or there's something going on in your chest. And on CT scan, what you see here is this large mass in the right chest, which turned out to be a relatively benign thymoma. That was resected. He did fine. And 18 months later, he developed COVID, did fine with that. No problems that anybody knew of, kept working throughout that. I didn't tell hospital epidemiology about that, but he was fine otherwise. But a few months after that, he developed progressive dementia. And by the time he was referred here, you can see in December, he already had more atrophy than a 40-year-old ought to have. And by 6 months later, he had profound atrophy and dementia. And brain biopsy showed dramatic accumulation of vaccine strain yellow fever virus. And both plasma and CSF had autoantibodies blocking interferon alpha, common in thymoma. And this patient, who was taken care of by Farinaz Safavi, we were not successful in getting him through. But we're trying to learn from this about what is it that leads to these antibodies and why are they so important in thymoma and what's the difference between those who get sick and those who don't. Okay, so I'm really done here. I wanted to try and give you an overall perspective of some of the things that we've been working on for the last 20 years. These antibodies are involved in a whole host of activities around how macrophages and T cells interact, as well as macrophages and many other cells. And I've only spoken about these here on this side of the diagram. There are many other autoantibodies that I've not touched on, and I've been paid to not do that. But these are important things that modify disease, and they are often not sought. So these anticytokine autoantibodies, they mimic Mendelian defects. We've talked about interferon gamma, Gm-csf, interferon alpha. These are really involved in a variety of things, and sorry, I left off IL-23, a novel autoantibody that is an important disease modifier. There are a lot of remaining questions here. What are the triggers for these antibodies? What are the predisposing risks? We talked a little bit about HLA. How long do they persist, both before and after? You looked at Gm-csf, which can last for years before, but I don't think that's the case for some of the interferon alpha cases, and probably not for some of the interferon gamma. Do they need to be eliminated? Not all of them do. Maybe life is better if we do, but we don't always have good mechanisms for that. And then finally, why do different diseases have different patterns? There are several diseases that all share type 1 antibodies, anti-interferon alpha, but they're different diseases, and they have different spectra of autoantibodies on top of the interferon alpha. So these are acquired innate and adaptive immune defects. They're dynamic. They can have on-target and off-target effects. They can be treated, and I'm absolutely confident that they affect more diseases than we think. So I'll just conclude. I've tried to acknowledge the people doing the work as I've gone along. Some that have really been key at every step of this operation include our collaborators in the Dental Institute, in particular Nikki Matsopoulos, our collaborators in Athens, Harry Matsopoulos, and our collaborators in the Arthritis Institute, in particular Sarthak Gupta. And I'll stop there. Thank you very much. I'd be happy to take any questions.
Everyone is free, or anyone is free to use the mics in the hallway there. I'll start with one online, if you don't mind, Dr. Holland.
Don't mind.
Okay. From our good friend Howard Young. Have you seen patients with antibodies of both type 1 and type 2 IFNs? If so, are their levels of type 3 high?
Yeah, Howard, that's a terrific question. And it's a fascinating one because in general, it's dichotomous. If you've got antibodies against type 1 interferon, you don't have antibodies against type 2 and vice versa. We really don't see much overlap between those. The type 3 antibodies, I got to go back and remember. We have seen type 3 interferon antibodies, but I don't know that we've got a clinical correlate for that. But the type 1-2 dichotomy, that's really quite fascinating to me, and I don't understand it yet. Dr. Matsoupas.
Yes. Dr. Holland, I'm not sure if this is on.
I can repeat it.
Am I loud enough?
Yeah,
So first, thank you very much.
Do repeat it, though. Yeah.
I will repeat it.
Well, I have a complex comment, but he can. So first, it was a treat, and it was very thought-provoking. Before asking my science question, I wanted to make a comment, and the comment was that it was wonderful to see your first slides of your path and the people that made a difference for you. And I wanted to make the comment that for many of us in this room and elsewhere, you are the person in that slide that has opened the scientific doors of exploration for us. So I just wanted to take a moment to say that.
Thank you.
My question comes related to IL-23, which is obviously something that I care about. Why do you think, this is a pre-40 antibodies, why are they specifically targeting mates that make gamma? Especially when the majority of mates that we know of in most of the barrier surfaces are 17 kind of type flavor responses, is the first part. And the second part is, are these mates that you looked at from the blood or from mucosal tissues?
Yeah, thank you, Nikki. So so why are they targeting gamma and not 17? I'm not sure that they're not targeting 17. I think 23 inhibition will block 17 production. It's just that, and perhaps it's our prejudice, that the gamma role is likely to be much more important in terms of mycobacterial disease, fungal disease, other intracellular infections. We know that 17 blockade is relatively common in APICED and other conditions, and I think the role for 17 blockade in disease pathogenesis is still somewhat unclear. In terms of, have we looked at mucosal tissue? No. And I think that, of course, we're always interested in outstanding collaborative opportunities, but we have not done that, and I think we probably should. These patients, many of them were recognized because this was all work done during COVID. Much of it was done on archival samples. And so we didn't have the luxury that we've had for some of our other patients where we had the patients here as the data have been evolving in real time. Now that we're back, we would love to engage our thoughtful collaborators in further evaluation of mucosal tissues. And I think by mucosa, I'm sure you mean gingiva, and we would be extremely enthusiastic about looking at that.
Okay, I have another one here. Maybe it's a consult your physician type question. Is there any general pharmaceuticals or special specific diet that Dr. Holland may suggest to lower autoantibodies in general post-COVID?
I've been trying Reese's peanut butter cups. So far, I'm feeling pretty good.
Mm-hmm.
No, I don't know about diets and how they might be effective in autoimmunity. We did hear a fascinating discussion this morning from Yasmin Belkade, who presented work regarding the effects of the ketogenic versus vegan diet and how that altered tropisms of immune cells. I don't think that those data are specific to how autoimmunity is generated, but I don't doubt that there could be dietary maneuvers, but I wouldn't know offhand what they are.
Good. Are cytokines important for maintaining a blood-brain barrier, or are the brain infections just a mass action effect?
Well, I don't know that I could answer whether cytokines are important for maintaining, or I should rephrase that. Of course, cytokines are important for maintaining the blood-brain barrier, as are so many things. And if the question is, are there cytokine dysregulatory syndromes that make for disrupted blood-brain barrier, I don't know. Certainly the blood-brain barrier is susceptible to inflammation, as are so many things. And cytokines are part of the inflammatory milieu. I don't think I have a better answer than that, except to say that cytokines are how cells talk to each other. And when they're upset, they sometimes say nasty things.
Okay, maybe similar. So any of the patients with autoantibodies to cytokines have variants in those genes? And also from Jay Hufnagel, are the autoantibodies IIG, IgM, or both?
Yeah, so the first question about are there variants in the target genes, that was our first hypothesis. And so we sequenced a lot of interferon gamma genes. Our hypothesis was that there would be variants in paternal interferon gamma that were being recognized by maternal immune response, leading to autoantibodies in young women in particular. It was a great idea that just happened to be wrong. And we've not gone to look for all the other cytokine targets. It's a good question. We've got now whole genome sequence on virtually all of these patients that we've seen here, and it is something that we will look at eventually, but I don't think that's going to be where the answer is. But it's a very good question. And then in terms of are the antibodies of the IgG, IgM phenotype, the answer is yes. There are some that, most of them are IgG. There are some that are IgM. Those, I think, are a little bit different in terms of the nature of the antibody and the nature of the response. But most of these are IgG antibodies.
Okay. From Steve Rosenberg, what are the reasons for the higher incidence in females?
Steve, that's a fantastic question that I think really gets to the heart of how little we understand about autoimmunity. And I'm an ID guy. I'm used to things that divide by fission, so I'm not really very good at these sort of dimorphic properties. And I think that what's so fascinating about this disease is that when you're in Southeast Asia, the rate of disease between males and females is like 60-40. Mm-hmm. When you get outside of Southeast Asia, it's 90-10. Why? So that really is that's not just an exposure. That's a later modifier. And I keep on thinking if we could crack that particular code, we would really be in a position to understand a lot about what is it that makes women have so much more autoimmunity. But just to put it in how I think we have simple stories that we tell ourselves, or I have simple stories that I understand. In the COVID case, the autoantibodies to interferon alpha are overwhelmingly male. And they're overwhelmingly in older men. So over age 70, the rate of these antibodies is really pretty high, a couple of percent. But women just don't seem to be getting it very much. So we have autoimmunity in the general that's higher in women, but autoimmunity to interferon alpha that's much higher in men.
Okay. Any thoughts as to the mechanism for the discrepancy between the titers and blocking EC50? Is it just that they are such high affinity antibodies and the treatment is not reducing the titers sufficiently to be effective?
Well, that's one possibility. I suspect what's really going on is that there are most of these are not clonal processes. They're not clonal populations of antibodies. They're not clonal populations of B cells. So we're dealing with polyclonal antibodies, and I think there are many of them that recognize different epitopes that we're not accounting for in the straight binding assays. We account for them in the neutralization, but not in the binding. And so, I think we can identify the changes in titer, the changes in neutralization, but we have to go beyond just single epitopes. And we need to develop better assays to do that.
Okay. Do you see epitope spread from binding to neutralizing determinants over time for any of the anti-cytokine antibodies?
I don't know the answer to that yet.
Okay.
I should, and other people, in the work of Chunlong Q, they do see epitope spread. We have not done that ourselves.
Okay. 3 o'clock, still holding up? Okay, you good?
I got my water.
Keep on going? Okay.
No obligation for anybody else.
Yeah, lots of questions still here. So can anti-IL-23 autoantibody in the P40 subunit also block the functions of IL-12? Have you found any autoantibody to the IL-23 P19 subunit? That's from Jeff Zhu.
Yeah, we haven't found blocking antibodies to P19 that I remember, certainly not clinically important. The blocking antibodies to P40 do block IL-12 as well as IL-23. Yes.
Okay, here was something I was thinking, actually. Have you ever looked in lupus patients? And might patients that do have the anti-type 1 IFN antibody have mild disease?
Yes, that is that Sarthak Gupta?
No,
So
it's our buddy Howard Young
that is
again.
a beautiful paper that Sarthak Gupta and Sarah Brown wrote in 2016 that was exactly that. So there is a relatively high rate of autoantibodies to the type 1 interferons in lupus. And those who have autoantibodies against the type 1 interferons have milder disease. Conversely, there's a relatively high rate of autoantibodies to interferon gamma in lupus. And those people who have autoantibodies to interferon gamma have more severe lupus by some laboratory criteria. In part because when you block type 2 interferons, you actually have more signaling through the type 1 interferons. So I think that it's exactly what was being suggested there.
Okay. I hear about patients who develop long COVID after multiple COVID infections who had few problems after the first or second infection. Is there a role for anticytokine antibodies in these patients? Could blood samples be obtained from such patients by a large collaboration?
The simple answer is I don't know yet, but there are several groups looking at this. I think it's a very important question. But let me just say that they will likely be different antibodies. So things that cause profound fatigue and mental fogginess and so on may not be the same antibodies that we're seeing that cause these infections. And that's fine. As you see in that work from PJ Utz, people develop a whole bevy of autoantibodies, not all against cytokines in the post-COVID setting. And so there may well be important autoantibodies that are related to symptoms that we're not going to pick up in our screen. Doesn't mean they're not there.
Okay. I don't know if you answered this, particularly part 2. Have the autoantibodies in various infectious diseases been cloned? And if so, what is the mutation rate?
Not by me, and I don't know.
Okay. Sonia Best asks, are the peptide sequence similarities, homologies seen in the first IFNG example combined with haplotype and opportunistic pathogens also observed between COVID-19 proteins and anti-type 1 IFN antibodies?
The anti-type 1 antibodies are not as HLA restricted as the anti-interferon gamma antibodies. No.
Okay, sorry, the font's kind of small here. Can immunizations trigger anti-cytokine antibodies?
I don't know, but so far we've not seen that.
Other cases of dementia following yellow fever vaccine? Question mark.
I'm sure there are. I don't know that they're related to yellow fever vaccination.
Okay.
I think this is a rare complication of yellow fever vaccination, but what I can't tell you is whether his interferon alpha antibody was from his thymoma alone or it was thymoma plus the COVID that made it happen. And I'm a little bit inclined to say the latter, because patients with thymoma and anti-interferon alpha autoantibodies often have more severe disease. And his COVID was actually pretty tolerable.
Okay, we'll end it with here. Wanjun Chen. Great talk. If I'm not mistaken, there is huge amounts of anti-IL-23 autoantibodies in the patients with thymoma. What might the underlying mechanism be, and are there any other autoantibodies against other cytokines in those patients?
Well, if I told you all that, I wouldn't have anything to talk about next year.
I see. Right.
So I don't know.
Tune in. Okay.
But we're trying to find out. So I think this is a broad question. What else is going on in these settings? How can we take an unbiased look at autoantibodies? And this is a broad question here. We know the things that we look for, but we're always at risk of only finding what we seek. And so we're trying to take less biased looks at these antibodies right now, especially in patients with thymoma, using more, entire holoproteomic approaches to see if there are other autoantibodies that might explain even more granularly why some of the manifestations are different.
Okay. Dr. Gilman, any closing thoughts?
Just I want to thank Steve for an outstanding lecture, and as the 25th in this series, I think it stands up with any and all of the best. And so thanks to you, and thanks to the audience. Thanks for everybody that submitted questions. I think it was a great session, and it's good to see everybody back in Lipset.
Sure is. Thank you very much.